a sprig of wild lettuce on a stone table. 3,000 years ago, a people about to flee their bondage were given 3 things to eat at their last meal as slaves: roasted lamb, unleavened bread, and a small, bitter green leaf pulled from the desert just outside the door. The lamb was sacrifice, the bread was haste. The bitter leaf has been called a great many things across 30 centuries. A symbol of slavery, a taste of memory, a relic of pastoral diet. It was also almost certainly a kind of medicine. Tonight, an investigation. We are going to look at the bitter herbs of the Passover meal, Marorim, in the Hebrew, not through the symbolism that the rabbis would later give them, but through the chemistry of the plants themselves. And we are going to find that the 5 species the Mishnah eventually named as acceptable for Maror share a single pharmacological signature. A signature that the body, 2,000 years before anyone could measure it, was already evolved to recognize. The argument is not that Moses prescribed medicine. The text of Exodus offers no medical reason for the bitter herbs at all. The argument is something subtler and, to my mind, more interesting. That when the rabbis of the 2nd century codified what counts as Maror, they selected plants that the Greco-Roman world around them universally recognized as digestive medicine. The convergence is not Mosaic and it is not modern. It is rabbinic. It is Hellenistic. And it sits, hiding in plain sight, on every Seder plate that has ever been set. The command itself is 1 verse long. And they shall eat the flesh that same night. They shall eat it roasted over the fire, with unleavened bread and with bitter herbs. Exodus chapter 12, verse 8. The Hebrew word for those bitter herbs is marorim, a plural, with a root that simply means bitter. There is no recipe. There is no species list. There is no medical claim. The text presents the bitter green as one of 3 elements of a meal eaten in haste, on the night of departure, in homes marked at the lintel with the blood of a lamb. The bitterness is, on the surface of the text, just a fact about the meal, not an argument about it. The command is repeated almost word for word in the book of Numbers, chapter 9, verse 11, the second Passover instruction, given for those ritually unable to keep the first. Lamb, unleavened bread, marorim. The triad is non-negotiable. The bitter herb is not a side dish. It is structural to the rite. What the Hebrew Bible does not do is explain why. That explanation arrives much later. In the book of Lamentations, chapter 3, verse 15, we meet Marorim again, but no longer as a food. He has filled me with bitterness. He has sated me with wormwood. Here the bitterness is metaphysical, the bitterness of exile, of the destruction of Jerusalem, of a national soul under judgment. By the time of Lamentations, the bitterness on the tongue and the bitterness in the historical memory have begun to flow into one another. And by the rabbinic period, the bitter green will have become, definitively, the taste of slavery. Rabban Gamliel, in the Babylonian Talmud at Pesachim 116a, makes this explicit. This bitter herb that we eat, for what reason is it? Because the Egyptians embittered the lives of our ancestors in Egypt. It is a flawless piece of theological exegesis. The Hebrew verb is the same root. The bitterness in the mouth becomes the bitterness in the body of the nation. The food becomes a sacrament of memory. This is the dominant reading of Maror, and it is the right one. It is what the herb means. What it does is a separate question. And to ask it, we need to leave Exodus, leave Lamentations, and walk into a much later text, the one where Israel's ritual met Greco-Roman pharmacology. Around the year 200 of the Common Era, the Rabbi Judah, the prince and his colleagues finished a great work of legal codification, the Mishnah, the first written compendium of the oral Torah. Buried inside it, in Tractate Pesachim, chapter 2, Mishnah 6, is a sentence that botanists will spend the next 1800 years arguing about, and these are the vegetables with which a person can fulfill his obligation to eat bitter herbs on Passover: hazareh, endives, chervil, field eringo, and maror. 5 plants, named in Hebrew, each one a real, identifiable growing thing, and yet each one, save the first 2, the subject of genuine scholarly dispute. Hazareh is the easy one, universally identified from medieval commentary through modern Israeli botany as Lactuca sativa, the cultivated lettuce, the same plant that sits in every supermarket today. Ulshin, endives in some translations, is Cicorium intybus or Cicorium endivia, chicory and its close cousin, also uncontested. After that, the trouble begins. Tamcha, the third on the list, was rendered as horseradish in the European Ashkenazi tradition, and that translation became standard on most Seder plates in the West. But Armoresia rusticana, horseradish, is a plant of cold European latitudes. It was unknown in the biblical and Tanaitic Levant. Modern critical scholars Zohar Amar, Yehuda Felix, Michael Zohari argue almost unanimously that the Mishnah's tamcha must have been something else, a wild, bitter chicory perhaps, or a sow thistle. The horseradish on the modern Seder plate is an exile-era substitution that quietly outlasted the original. Charchavina, the fourth, is traditionally rendered as field eringo, a thorny, thistle-like plant of the Apiaceae family. Zohari, looking at the Mishnah's clear preference for plants that bleed a milky, bitter sap from their cut stalks, has suggested instead that Charchavina names a second variety of chicory. The dispute is unresolved, and Maror itself, the eponymous bitter herb at the end of the list, has been read as wild lettuce, as sow thistle, as the poppy-leaved Ricciardia. We do not know with certainty what the first Israelite ate. Ancient cultures named plants by what they did, not by their genetic lineage. The fluid folk taxonomy of the Bronze Age does not map cleanly onto Linnean species. What we can say with certainty is this: 4 of the 5 species, Hazareh, Ulshin, Charchavina, and Maror itself on the modern critical reading, belong to a single botanical family, the Asteraceae, the daisy family. And the apparent outlier, tamcha, when restored to its likely Levantine original, sow thistle, Sontius oleraceus, is also Asteraceae. 5 plants, 1 family, 1 chemical fingerprint. The Mishnah, almost certainly without knowing it, had assembled a pharmacological set. The bitter principles in the Asteraceae are called sesquiterpene lactones. They are produced by the plant in latex-bearing channels that run through its leaves and, especially, its stalks. Cut a mature wild lettuce at the base and a milky white sap will emerge from the cut surface. That sap is the medicine and, simultaneously, the toxin. The 2 compounds that carry most of the bitterness in lactuca are called lactucin and lactucopicrin. The Greeks named the dried latex of wild lettuce Lactucarium, the milky one, and used it for 2,000 years. Pliny the Elder, writing in the 1st century, in Book 20 of his Natural History, called bitter lettuce amara lactensk, the bitter and milky, and listed 2 principal uses: as a sleep-bringer, somno, and as a remedy for stomachis dissolutis, what we would now call gastric upset. The Mishnah's instructions for Maror, written within the same Roman world, contain an almost uncanny detail. The Mishnah specifies that the herbs must not be pickled, must not be overboiled, must not be cooked. And the obligation, the Mishnah says, can be fulfilled by eating their stalk. Both stipulations preserve the very molecules that the plant uses to defend itself, the volatile, heat-sensitive sesquiterpene lactones that concentrate in the latex of the mature stem. The Mishnah did not know about sesquiterpene lactones, but it knew perfectly how to keep them intact. The Babylonian Talmud in Tractate Pesachim Folio 39a notices something else. The rabbis observe that lettuce is sweet in its early growth, the soft, mild young leaves, and bitter in its mature stalk when it bolts to flower. They read this biologically and historically together. Just as the lettuce begins sweet and ends bitter, so did the Egyptian exile begin sweet under Joseph and end bitter under Ramses. It is a metaphor. It is also a phytochemistry lesson, because that is exactly what lactuca does. When the plant transitions from vegetative growth to flowering, under heat, under stress, under age, it dramatically increases the production of lactucin and lactucopacrin in the stalk. The mature, bolted lettuce is up to 50 times more bitter than the young leaf. The rabbis described the life cycle. Modern phytochemistry describes the mechanism. They are looking at the same thing through different vocabularies. Chicory carries a parallel set, cichoric acid, plus its own family of sesquiterpene lactones. The European Medicines Agency, in its herbal monograph on Cichori radix, chicory root, recognizes traditional digestive use formally enough to grant it a regulatory category. Horseradish is the outlier in the modern Ashkenazi list. Its bitterness comes not from sesquiterpene lactones, but from a sulfur compound, sinigrin, which releases allyl isothiocyanate when the root is grated. A different family, a different chemistry, but, as we will see, not a different receptor. In the year 2000, in the journal Cell, 6 researchers from the laboratory of Charles Zucker at Columbia University published a paper that changed our understanding of taste. Adler, Hoon, Mueller, Chandrasekhar, Reiba, and Zucker described for the first time a family of mammalian receptors dedicated to a single perceptual category, bitterness. They named the family TAS2R. The original count was around 24. In humans, the family has settled at 25. Each TAS2R is a G-protein-coupled receptor, the same broad architecture that handles adrenaline, that handles serotonin, that handles smell. The TAS2Rs sit in the membrane of specialized cells, threaded through it 7 times, with a binding pocket facing outward. When a bitter molecule docks into that pocket, the receptor changes shape on the inside of the cell, releases a signaling cascade, and the brain, eventually, perceives bitterness. For the first several years after their discovery, the TAS2Rs were assumed to be a tongue-only system, a specialized sentinel against ingesting plant toxins. That assumption did not survive the next decade. By 2010, the TAS2Rs had been found on enteroendocrine cells in the lining of the stomach and the small intestine. By 2015, in the smooth muscle of the human airway. By the early 2020s, on circulating immune cells. The taste receptor was not a taste receptor at all. It was a chemosensory system expressed across the body that the tongue happened to use as one outpost. What it does in the gut is, in cell culture and in rodent models, beginning to be mapped. In 2011, Inge de Portere's lab in Leuven published an anchor paper in PNAS, Janssen and colleagues. They showed, in mice, that activation of bitter taste receptors on enteroendocrine cells triggers the release of ghrelin, the hormone that signals hunger and modulates gastric emptying. Subsequent work in human intestinal organoids in cultured enteroendocrine cells has added GLP-1 and CCK to the list of hormones that bitter receptor activation can mobilize. And alongside the hormonal cascade, oral bitter exposure also primes the cephalic phase of digestion through the vagus nerve, the parasympathetic nerve that runs from brainstem to gut, the same nerve that fires when you see and smell a meal before you taste it. So far, the script could write itself. Bitter compounds in the Mishnah's 5 plants activate the bitter receptor family. The bitter receptor family is expressed throughout the gut. Activation of those receptors releases digestive hormones and triggers a vagal cascade. The Mishnah, by accident, ratified a pre-meal medicine. This is the line the science will not yet let me cross, because the careful work, the systematic reviews, say something more cautious. The 2021 review by Rezaie, Bitarafan, Horowitz, and Feinley-Bisset in Nutrients puts it plainly. The evidence that bitter substances affect gastrointestinal function in healthy humans is limited, inconsistent, and largely restricted to studies using pharmacological concentrations via post-oral routes. In English, it has been tested with feeding tubes and capsules and isolated compounds, not with a few grams of lettuce on the tongue. No randomized trial has measured what a Seder portion of Maror actually does to gastric acid, to bile flow, to the cephalic insulin response in a healthy adult. The receptor biology is real. The cellular cascade is real. The leap from the receptors exists in your gut to eating 14 grams of lettuce primes. Your digestion is, today, an open scientific question. We don't know. The work has not been done. What we do know is that the rabbis chose plants whose bitter principles activate this system. Whether the Seder portion is enough to move the needle is a question only physiology, not history, will answer. There is one more detail worth pausing on: the architecture of the Seder itself. The Seder opens ritually with karpas, a green vegetable, often parsley or celery, dipped in salt water. Then the bitter herbs. Then, eventually, the festal meal, with the roasted lamb at its center. Karpas, then maror, then meat, a small bitter green, then a stronger bitter green, then heavy protein. In 1984, the scholar Baruch Bokser argued in The Origins of the Seder that this sequence was a Jewish appropriation of the Hellenistic symposium, the Greco-Roman dinner party ritual, in which bitter and astringent appetizers preceded the heavy courses, intended in the medical theory of the day to stimulate digestion. Joseph Tabori has pushed back. Dipping greens into salt water was a common practice across the ancient Near East, not invented from a Roman template. They are probably both partly right. The rabbis took an old Near Eastern practice and stylized it into the form their world recognized. Whatever the ancestry, the timing is striking. The bitter is at the front, the protein is at the back. And in the moment of consumption itself, Hillel the Elder is recorded in Pesachim 115a, as insisting that the matzah, the lamb, and the bitter herbs be eaten together, wrapped in a single mouthful, to fulfill the literal sense of the verse. With unleavened bread and bitter herbs, they shall eat it. What this means physiologically, if it means anything physiologically, is that the bitter exposure happens at the precise moment of swallowing, the exact moment at which oral bitter receptors fire most strongly. The script will not claim Hillel knew what he was doing in a pharmacological sense. He almost certainly didn't. He was a halachist, not a physiologist. But the ritual he insisted on, 2,000 years before Sell published Adler and Zucker, was a ritual that puts a bitter agonist on the tongue at the optimal moment for receptor activation. The medicine timing matches whether the rabbis intended it or not. It is tempting at the end of this kind of investigation to overshoot, to claim that Moses encoded modern science or that the Seder is a hidden pharmacology disguised as a religious meal. Both claims are false. The text of Exodus does not give a medical reason for the bitter herbs. The rabbis read them as memory of slavery, and that reading is the right one for understanding what the herbs mean. What the herbs do is another question, and the convergence we have found is not between Moses and modern biology. It is between the rabbis of the 2nd century and the physicians of their own Greco-Roman world. The Mishnah selected 5 species that Pliny and Dioscorides and Galen, working in the same generation, classified as digestive medicines. The rabbis were not encoding pharmacology. They were ratifying a folk botany that their surrounding medical culture already understood. 2 intellectual traditions, looking at the same plants, calling them by different names, Merorim in Hebrew, Emara lactensk in Latin, and ending at the same 5 jars. 2 millennia later, a paper in Cell described why. The bitter compounds in those plants bind to receptors that the human body expresses, not just on the tongue, but throughout the gut and the immune system. Whether a Seder portion is enough to do anything physiological in a healthy modern adult, we do not yet know. The science has not gone that far, but the convergence at the level of biology is established. The rabbis, in the act of building the ritual that would carry their people through exile, chose the plants the physicians of their era prescribed. The bitterness on the tongue, once meant simply to soothe the stomach, has been permanently repurposed to agitate the historical memory. Both meanings are real. Neither cancels the other. This is Remedia Sacra. If you'd like more of these, the subscribe link is in the corner. Thank you for watching.